Hi everyone, welcome back. So today I'm going to do the extension of my first video, which was on um, Orthofinder and how to run Orthofinder, where to find the results. So today I'm going to talk to you about how to interpret those results and how to download those results onto your local. So once again, please check out the written tutorial. This is going to have all your information on how to use it, what it's about, and parameters you can use with it, as well as the output. So I'm going to get into the results. If you would watch my previous video, you'll see that this is where we left off. We have the Orthofinder results right here. So all these directories we really want to take a look at, and the best way to do that is to download it onto your local. So to start, I'm going to log out of my terminal. Once again, if you're not using a cluster, you don't really have to worry about this. It's already going to be on your local. So for those of you who do have clusters, this is how I take care of this. So I'm going to log out first. I'm going to use the secure copy command again. This time, since I'm specifying a directory, I'm going to use that recursive parameter, so -r. And since I'm trying to get a directory that's already on my cluster, I have to start by specifying my cluster account and the pathway to that directory. So let me just write that out. And you can see that the pathway to that directory is right here. So shared virus Orthofinder analysis, Orthofinder, and then results. So I'm actually just going to copy from virus on here so that I don't have to write it out. Being a little lazy here. Now I got to get rid of that double slash. There we go. All right, so that's the directory that contains your results. Now I want to specify where I want to put it. So I'm going to put those right onto my desktop. That would be users, David Blumsack, and desktop. Now it's going to ask me for the password to my cluster account. I think I just messed up that password, but hopefully not. All right, so there's going to be a lot of files here. Shouldn't take too long to download them all, though. And when you download all these, it maintains its structure in the directories. So it's not like one folder that's going to contain all these files that you have to make sense of. They're still going to be in their directories, such as comparative genomic statistics, orthogroups, and so on. So you can see it's already finished. That's because I only had 3 proteomes. As you get bigger proteomes and more proteomes, it might take a bit longer, and you might want to just download individual directories instead of the whole thing in order to make sure that it downloads quicker. So I'm going to go to that now. It's on my desktop, and it should be called results. So results right here. Nope, that's not it. Right here. So this results folder has all my directories or folders that were on the command line. And let me just go back and show you. All those folders represent these blue lines, which represent directories. So every single one of them now has its own folder right here. And now I can look at each of them and open them up on my computer. All the .tsv files, tab separated, can be viewed on Excel. So I'm going to get started with the comparative genomic statistics. And I'm not going to go through every single one because that would take an extremely long time, but I'm going to go over the ones that are most useful, in my opinion. So right here, you have statistics overall and statistics per species. So let me open that. Sorry, old stuff. Statistics overall. So this is broad information about your whole Orthofinder analysis. The number of species you used. So I had 3 viruses that I'm using, SARS, MERS, and SARS-CoV-2. So that's 3. The number of genes among them, 116. The number of genes that were actually assigned to orthogroups, and orthogroups are just groups of sequences that are similar and they might represent a individual protein family or gene family. So that's 104. And a lot of this other information is extremely useful. Down here, you can see the average number of genes per species in orthogroup. And then number of species in orthogroup. So there were 15 orthogroups with all 3 species in it. That's extremely useful information, especially if you're looking to see how widespread a gene is in a group of species or distantly related species. And then 9 of those orthogroups had 2 species in it. And I mean, that can help you identify differences among the viruses. There's definitely differences and similarities. So if one of the viruses isn't in the ortho group, there could be differences that you can build off of from there. And now I'm going to get into some of the other results. So let me go back to the folder here. Statistics per species. So right here, you can see these are the 3 proteomes that I used as input. It's specifying them right here. And now it's just going to give you broad information about these proteomes individually. So for example, the number of genes in the SARS proteome was 41. Number of genes in MERS was 37, and so on. And then it will give you other information, obviously, like unassigned genes, percent of genes in ortho groups. And once again, that's like broad information that you can use just to understand how good the Orthofinder analysis was, what it was able to cover, and so on. And then how might even give you information on like the quality of the proteomes. If a lot of genes are matching and or you think that a lot should be matching and they aren't, there could be things that you could look into from that. And then here you can see number of genes per species in ortho group. So that's just broad level information. I'm going to get into some of the more specifics now. So this is just comparative genomic statistics. It's just one of the many folders that it outputs and many results that it outputs. Those are the 2 folders or the 2 files that I find most helpful in this directory. I'm not going to get into the others, but certainly explore them on your own time so that you really get a good understanding of what Orthofinder outputs as results. Next, I'm going to go into gene duplication events. So that's another TSV file. And right here, it's a bit hard to interpret on Excel, but it's just going to give you information on possible duplication events that have occurred that may have led to, you know, the phylogeny of the genes that it understands. And this, it's a bit hard to understand on Excel, and it's not one of the things that I find as useful through Orthofinder, nor do I find that it, you know, it's just all hypothetical. But it's just supplementary information. Another thing you can look at, and looking at some of the trees and the gene trees, species trees that it outputs along with this information can help give you an idea of like the evolutionary origin of some of these genes throughout those three species of, or three viruses. We're only looking at three, so it's not going to be as useful, but if you're looking at a lot of different proteomes, then that becomes a bit more interesting. So back here, I'm going to go into gene trees. Now these, I don't have a tree viewer to look at them at the moment, but if you have a tree viewer, you will be able to see all these trees and just look at, for each individual ortho group, basically, it creates a gene tree that just shows you the descent of each of the genes from each of the species in that ortho group. Once again, it's not really as easily readable without a gene tree, so I'm not gonna get into that. It's fairly simple anyways. Right here, orthogroup sequences. So out of all these orthogroups, so Orthofinder created 35 orthogroups for these three viruses, and then all these files contain the sequences that are in each of the orthogroups. So every sequence that was lumped into orthogroup 0 is right here, and so on. And that's just pretty basic right there. Going into the orthogroups, there's another important TSV file here, orthogroups right here. Looking into that, it's going to tell you the orthogroups, and it doesn't show you all of them, it probably just shows you the ones that have either 2 or 3 species in it. So it specifies in this first column the ortho group, the second column, third column, and fourth column, it specifies the species or the virus or the proteome that you use as input. And you can see under each of these, it shows you the sequences from that proteome that are in this ortho group. So in ortho group 0, these proteins sequences are these sequences from SARS are in that ortho group, and so on and so on. So this is really good information to know. You can take these sequences individually out and maybe look at them, as in alignments. For ortho group 0, you might want to look at the alignments of these proteins and so on. So that's just good information to know what sequences are actually in there and how they're distributed and what ortho groups have what. So that's another important file to know about. And there's other files in here that give you gene counts and unassigned genes. I'll look at gene counts as the other one. And this is kind of like just adding on to the orthogroups file. It just tells you the number of sequences from that proteome in that orthogroup. So if I go back to that one called orthogroups, each sequence is separated by comma, so you can see 1, 2, 3, 4, 5, and going back to here, it says 5. So it's kind of the same information, but it's a little bit easier to see how many are in each, but it also gives you a total, which is cool. So going back to the results, unassigned genes would just be the genes that aren't assigned to orthogroups. I'll go on to the orthologs now. So it gives you orthologs for each of the viruses or the input individually. So if I go here, gives you orthologs that were found. I can go into here. So, for example, here in orthogroup 0, it believes that these sequences right here from SARS and MERS are orthologs, and so on. And it gives you that information for all of your input. And that's just another interesting thing to look at, and it could help give you an idea of how these genes have evolved. And I mean, it's just cumulatively, all this information together can give you a really good grasp on, the origin of these genes, and also, just even what they function, because you can look at how other proteins that might be similarly grouped function. So going back to here, going back to results, you next have phylogenetic hierarchical orthogroups. And this one, I have to say, it's one that I don't necessarily look at as much. What it gives you is orthogroups found using a method different from the gene similarity method that produced the results in the orthogroups directory. And so these orthogroups are just, I believe they're more accurate, and they provide additional take on the relationships. But I personally don't use it as much. I think that the information here is it's much more abundant and more useful, in my opinion. But yeah, I would definitely recommend exploring the manual from David M's on his GitHub repository, and just reading up about all of these just to get an even better idea. Like I said, I'm just giving you a broad overview of this. So for putative xenologs, this just contains all the information pertaining to xenologs predicted by Orthofinder. And xenologs, if you don't know, are orthologs where homologous sequences are found in different species because of horizontal gene transfer. So that's interesting, especially if you're looking at things like bacteria, because horizontal gene transfer is more common among them. I'm not going to get into that. It's just more information you can take a look at. Then resolved gene trees. They're just gene trees again. I can't look at those because I don't have a tree viewer. So these are all in text format, but they can be seen using a tree viewer. Next, single copy ortholog sequences. This just contains all the sequences that belong to orthogroups which contain only a single sequence from your input. So it lists the orthogroup that has the information, and then it just contains the sequence as well. And that's just a FASTA file. Next, it does produce species trees as well. Again, I can't look at these, but it's just good that you know that it does produce species trees. These types of trees you can also make using like any tree like software, tree making software like Genius. And I haven't yet compared the output of the trees. I'm hoping that they're very similar because I want to trust these, but it might be cool to take a look at how the trees are made among different softwares and see if they're like that much different. I'm not sure the exact method that it makes these trees. That can probably be found in the manual as well. Lastly is the working directory. This is like all the information that it produces, all the results that it produces to get to the results that it has in here. If there's a problem in running Orthofinder, this could be a useful place to look to see what output it doesn't have, or if there's any error created. There's also citations and logs. So that was just a really brief overview of the results that Orthofinder outputs and how to even get them on your desktop. Again, if you go to this website here, this is the GitHub repository for Orthofinder, you can find out much more about this information. And this manual right here, it goes through installation, and it also tells you what Orthofinder does and the exact output that it produces. So results file, orthologs directory, ortho groups directory, gene tree, resolve tree, so on. I also touch upon this briefly in my written manual, so if you want to see a dumbed down version, you can check that, but this is going to have the most information for you. So if you really want to know the specifics about how Orthofinder works and the results that it outputs, I really suggest giving this a thorough look and getting a good understanding of how it all works. So thank you for tuning in for this video. That's all I have for you, and thanks.